Respected Dr. Devanshu Patel, sir, Honorable President, Parul University, Professor M.N. Patel, sir, Honorable Provost, Parul University, Dr. Abhay Dharamsi, sir, Dean, Faculty of Pharmacy, Dr. Vandana Patel, Madam, Dean, Doctoral Study, Dr. G.S. Chakravarti, sir, Principal, Parul Institute of Pharmacy and Research, Dr. Lalit Jha, Madam, Principal, School of Pharmacy, and faculty members and all the participants and attendees. Very good morning to all of you. In the continuation of our webinar series, today we are here to take part in all-inclusive webinar on the title Nanomedicine and Nanobiotechnology, History, Progress and Challenges. For this talk, we have a special guest, Dr. Rishi Paliwal, sir. He is an expert of this area and also he is a motivational speaker. So let me introduce to all of you about sir. Dr. Paliwal is presently holding a position of Assistant Professor in Department of Pharmacy, Indira Gandhi National Tribal University, Amarkantak, Madhya Pradesh. He has completed his bachelor, master and doctoral degree in pharmacy from Department of Pharmaceutical Science, Dr. Hari Singh Gaur Vishwavidyalaya, Sagar, Madhya Pradesh. Dr. Paliwal has completed his postdoc from Rangel College of Pharmacy, Texas, AM University, USA. Dr. Paliwal is a recipient of Young Scientist Award given by Madhya Pradesh Council of Science and Technology, Bhopal, for his research effort in the area of nanomedicine and nanobiotechnology. Sir has 14 years of teaching and research experience in the field of pharmaceutical sciences. And Dr. Paliwal is also a recipient of Rudolf Sindins Award given by European Society of Biomaterials, Career Award for Young Teachers by AICT New Delhi, and Outstanding Pharmacy Teachers in Pharmaceutics by APTI MP. His research area of interest include application of nanotechnological principles in drug delivery, drug targeting, lymphatic delivery of therapeutics molecules through oral route, non-invasive delivery of macromolecules such as protein and peptides, including vaccine delivery and intracellular drug delivery. He has published more than 40 research papers in high-impact internationally reputed journals, and he has contributed about 18 book chapters in leading international books of material science, nanoscience, and pharmaceutical science. Dr. Paliwal has recently written a book on advance in nanochemo prevention and controlled drug delivery of phytochemical bioactives with Springer Nature, Singapore. He is also editor of 2 upcoming reference books by Elsevier and Taylor of Francis in the area of targeted nanomedicines for breast cancer and advance in nanomedicine, nanotheranostics, and nanobiotechnology, respectively. He has participated and presented his research work in several international conferences in India and abroad. Dr. Paliwal served as a reviewer of many international journals, including International Journal of Nanomedicine, Expert Opinion on Drug Delivery, Biotechnology Advances, and International Journal of Biological Macromolecule. He served as a guest of editor of special issues of journals Current Drug Therapy and Mini Review on Medicinal Chemistry. He is editor, board member of the International Journal Current Drug Therapy, Bentham Science, published in 2016. At present, 3 research projects are going on, which are sanctioned by DBT New Delhi and UGC New Delhi, worth 75 lakhs. Dr. Paliwal has organized various seminars and conferences which are sponsored by DBT and AICT. And he has also delivered several invited talks in various national and international conferences and seminars. With this, I invite Dr. Paliwal sir to deliver his talk.
Thank you so much, Dr. Binod. My slide is visible. I think it is full screen now. Is it okay?
No sir, full screen नहीं है अभी.
Okay. Actually, I tried, but I don't know why it. I'm trying it because it is important. Is it okay now?
No sir, entire screen पे करिए sir इसको.
No, I did it entire screen. In my laptop, it is showing entire screen actually. Some issues there. Just a minute. Is it visible now?
सर वैसा ही आ रहा है अभी.
Okay, there is some issue. I don't know. मेरा लैपटॉप में तो फुल स्क्रीन दिखा रहा है. Still there is some issue.
आचार्य सर.
Should I continue like this? It may be some technical issue. I don't know.
आचार्य सर से पूछ लेते. आचार्य सर. हेलो आचार्य सर.
Just a minute. Okay. Is it visible now?
Yes sir. Yes sir.
Perfect. Thank you so much. All right. So sorry for this technological issue. Thank you so much, Dr. Vinod. And first of all, I would like to thank Professor Jha, Madam, and all the Department of Pharmacy at the Parallel University, and Dr. Vivek Srivastav, sir, and everyone, those who considered me as one of the possible speaker in this webinar series. I have little bit experience of working on nanomedicine and nanobiotechnology. That's why I have chosen this topic, history, progress, and challenges, because many of us are not familiar how the nanotechnology progressed in the country and worldwide. So I will try my best. Hopefully you will enjoy the lecture. So the outline of this lecture is that what is nanomedicine and how it is different from the conventional medicine, and how the concept of nanomedicine came into the existence, what was the history behind it, and how it progressed slowly with the advancement of the techniques and instrumentations, and what was the milestones and breakthrough, particularly in the researches of nanomedicines, and how the people got motivated to work in this direction, and what is the current status of the nanomedicine market, and what are the future trends for that, and how fast it is growing. So what are the common methods of developing a nanomedicine or nanobiotechnological tool, and what are the challenges for this nanomedicine development and its regularization, and whether there are some scale of feasibilities in the nanomedicines, and if they are, then what are the pros and cons. And also I'm going to highlight a little bit about how the nanotechnology merged with our Indian ancient culture of the herbal medicine, and how it is important in prevention of the cancers. And what are the latest upcoming concepts in these areas which are combining at the nanotechnological platform. So one by one I will start. So why the concept of nanomedicine exists, and why there is a need of small TB medicine, particularly for treatment of different diseases. So we can imagine a type of disease and a type of treatment. If suppose a doctor search out some very first cancer cell in the body, with the help of certain tool, so that there is no progression of the cancer in the body, and otherwise there will be a development of tumor. If usually it happens because we fail to add early diagnosis of any kind of disease which is dangerous at the later stage. Second, if we know that there should be some sort of nano robots which can go and correct the problem in the cell, either intracellularly or at the organ level or the tissue level, and to that only to that particular cell which has required treatment from that side. So that kind of concept came in the mind of the people, and these are the miniature biological machines actually. So they work in the body and they go and to the target site particularly and treat that particular cell only. Third important thing is that if suppose we imagine that if we pump the size of the molecules could be implanted to deliver the life-saving medicines, particularly at the target site, precisely because we don't want to expose the body with these dangerous drugs, whether synthetic or biological drugs. So how precisely we can deliver the drug at the target site, that is very, very important. So that was the all need, and this is only possible with the help of some small engineered machines that can perform actually in this direction. So nanomedicine is usually different from the other kind of medicine in many terms because they are very, very small particles. They cannot be visible with the naked eye, and they are the purposely designed actually nanocarriers for some better therapeutic and diagnostic purpose. Like we can imagine and we can see what are the nanoscale. So from 1 nanometer to 1000 nanometer we go and we see. So there are very, very tiny particles. In our body already there exist the nanostructures like hemoglobin is 6.5 nanometer, antibodies generally has 12 nanometer, even influenza virus is 130 nanometer, like coronavirus also having same size. And the dendrimers are very less size, less than 5 to 10 nanometer, 5 to 100 nanometers. There are range polymeric therapeutics, metal nanoparticles, block polymers, nanocrystal technology, polymer-based nanoparticles, and the suspensions, particularly nanosuspensions, liposomes. So there are n number of different type of nanostructures that can be developed from 1 nanometer to 1000 nanometer. So ideally that nanotechnology, the definition is that any particles which has at least one dimension in the range of 1 to 100 nanometer, that will be considered as a nanoparticle or nanotechnological feature. So that is what we are going for. So these are some of the systems. But from where this came in the mind, because nanotechnology, we know that it is applicable in other field also, and it is physical matters and other parts of the world. It is being applied in almost every part of the science and technology. So one paper was published in 1959 by a Nobel Prize winner physicist Richard Feynman. And that was very, very popular lecture actually, and very popular scope. However, there was no term nanotechnology or nano something. But that time, all research was going on the macro level, how large, how giant the material is, how big is the universe, everything. But that time he wrote a letter and said that there is a plenty of room at the bottom, and an invitation to enter a new field of physics. So he said, okay, we can go with the level of lower than the micron levels and the nano levels. There are possibilities. He has not used the word nano, but he says that there are some small particle world exist in the whole universe, and that is have its own law and regulations. That is not following the other physics law of that are existing. Their law are different. Their nature is different. Their phenomena are different. So this was the very big significant article. It attracts the attention of the many people in this direction. And then there was one book in 1986 written by the Eric Drexler, Engine of Creation. And he said the coming era of nanotechnology. So this was the book who just attracted the attention of the people. Okay, this is the area where we can work upon, and we can design or we can create a small engine, because electron microscopy has been developed and we can see these nanoparticles by that time slowly and slowly, so that we can understand their phenomena, and that was very much important. So first time if we talk about the the nanomedical device design technical paper was published in 1996 by Freitas, and the title was Respirocytes and Artificial Red Cells. So he imagined that if we can engineer some RVCs having extra capabilities of having additional ATPs and as a source of delivery for this kind of thing, it will enhance the performance of the athletes and the people who are living on high altitude area, etc., etc. So this was very important paper, and where nanomedical device design was proposed. And in 1991, again, there was one book that was on Unbounding the Future, the Nanotechnology Revolution by Eric Drexler again. And here it was the first time they used the word nanomedicine. So this was a little bit a time period when the people started thinking of it and started working in this direction and understanding the phenomena of the nanomaterials. So the overall nanomedicine, nanobiotechnology work in an integrated manner. If we see that nanobiotechnology has several specific applications in life science, in genomics, proteomics, in nanoarrays, molecular diagnostics, as in biomarker, cell and gene therapy, drug delivery, drug discovery, and all this together comes to the two part. One is nanobiopharmaceuticals and another is nanodiagnostics, if we talk about nanobiotechnology. And if we talk at the interjunction of nanomedicine, we see that all these three dimensions ultimately lead to the personalized medicine, because one individual has the different type of different type of response to the medicine, and that's why it should be customized from person to person. And this is what the new concept is. So ultimately leading to the personalized medicine, not the generalized medicine. So it was assumed that what should be a ideal nanoprobe. Because ideal nanoprobe, it was thought that it should have a very small size and which can pump the toxins from the body and also can dispense the drug. There should be some sensor which can sense the requirement of that particular cell, and there should be some manipulators that can detect illness and perform the cell to cell surgery. They might also have certain sort of for that surgery there should be cell make of some strong chemically inert material like diamond. And also they can relay the information on board on computers and send and receive the ultrasound to communicate with medical team, which is outside in the operation theater. So that has been imagined actually, that the ideal anatomy of a nanoprobe or a nano device that can work in this direction. So if I talk what was the milestones and breakthrough research in nanomedicine and particularly drug delivery point of view. So we know that at the very beginning of 20th century, the Paul Ehrlich proposed a concept of magic bullet. In magic bullet, they said that drugs should be added into such bullets, and these bullets should only deliver the drug at the target site, not the other part of the body. So this is still an ideal situation which was proposed very early by the Paul Ehrlich, and still we are trying to cope up with that. At the end of 1960s, the Peter Paul Spacer developed the first nanoparticle that is used for targeted drug therapy or targeted drug therapy. In 1970s, the George Jean Frank Kohler and Cesar Milestone succeeded in producing some monoclonal antibodies for particularly achieving this targeted drug delivery. At the start of the 1990s, nanoparticles were modified for the first time for the transport of DNA fragment or the biologics, we say, and genes, and they were capable of entering into the cells with the help of these antibodies. So by understanding the body physiology and developing the monoclonal antibody for the same and anchoring these monoclonal antibody on the surface of nanoparticle and targeting that particular problem in the cell and that particular gene where there is a requirement. Afterwards, we know that after 2000 onwards, the gene, nucleic acids, proteins, molecules, and cell processes can be rendered visible in real time with the help of certain advanced nanostructures like quantum dots. So diagnostic of the diseases and the cells was possible. Live imaging become possible. More and more advanced techniques are upcoming. And these nanoparticles of using some semiconductor crystals, they were also used, which can diversely modified electronically as well as optically, and they can the change in the light signal and other sensing signals, we can easily detect the problem in the body and the cell. So when we are talking so much about the nanomedicine, the question is what is the market for the nanomedicine and how fast it is growing? Because if we develop a nanomedicine in the lab, what will be the future? Who is going to buy and why the people are going to buy? Before 2000, the concept was to develop more and more conventional formulations and medicines. But afterwards, in 2000 onwards, there are significant increase in the market, and so many pharmaceutical companies are coming and they are developing many nanopharmaceuticals. So if we see that what is the trend in particularly only United States nanomedicine market as a product from only 2013 to expected up to 2025 in US billion dollars, you see there is an increasing trend. Almost it was started from 1.9 billion US dollar, and now it is expected in 2025 it will be at least 5 times more. And now you can see that the product are being developed in all the directions, including therapeutics, also for regenerative medicines, the diagnostics for in vitro diagnostic and identification of the problems quickly, and in vivo diagnostics as well, and vaccines also. So it is increasing, but major area is therapeutics because this is more easy regulated by the regulatory bodies, and however the demand is also more for therapeutics. If we talk about the product outlook in the terms of revenue, which area is creating more revenue in US dollar? So we talk about the therapeutics are the highest one, where if you are using some therapeutic molecule, if you are developing some therapeutic nanomedicine, it is more useful, more revenue based. Second is regenerative medicine, then in vitro diagnostic, in vivo diagnostic, and vaccines. If we talk about the applications part, where these nanomedicines is more applied, where is the patient and the consumer, if I say, they are ready to spend the money in terms of revenue. So clinical oncology, because the number of cases of the cancers are increasing day by day, and we need and we know that cytotoxic drugs are too much harmful and have several side effects and adverse effects in the body. So chemotherapy has their limitations. So that's why more and more the designed and targeted a nanomedicine product based on the cytotoxic drugs are being developed. So clinical oncology is the highest one, which is covering the revenue from the application part also and revenue point of view. And it is increasing day by day because we know that the number of cases are increasing. So clinical oncology is the highest one. Then after infectious diseases, then we go for clinical cardiology, and then orthopedics, and then other miscellaneous applications. So these are the areas where the market trend is going on. If we talk about certain product and their companies, I can give you some numbers so that you can understand the importance that, okay, there's so many companies are upcoming. So these are some of the name, like Blend Therapeutics, there is a nanoparticle based drug delivery. Then Crystal Therapeutics, they are working in polymer micelles nanoparticle for delivery of drug to tumors. Then Systemune gold nanoparticle for targeted delivery. DNA Medicine Institute diagnostic testing system. Invitrogen, they are using quantum dots for medical imaging. Luna Innovations, they are using buckyballs to block the inflammation by trapping free radicals. Then Makefield Therapeutics, they are using nanoparticle cream for delivery of nitric oxide gas to treat infections. Then Megare, they are using diagnostic testing systems. NanoBio, they are developing nanoemulsion for nasal delivery to fight viruses like flu, cold, or other through the skins to fight bacteria. Nanobiomagnetic, magnetically responsive nanoparticle for targeted delivery. Nanobiotics, nanoparticle that target tumor cells and eradicate the X-rays. The nanoparticles generate electrons and they cause the localized destruction of the tumor cells. And nanoprobes, that is gold nanoparticle for radiation therapy and enhancement. Nanospectra, they are using Orocells particle for thermal destruction of cancer cell. Nanosphere, they are using diagnostic testing using gold nanoparticle to detect the low level of protein indicating a particular disease in the body. Nanotherapeutics, They are using nanoparticle for improving the performance of the drug delivery by oral and nasal methods. And Oxonica, they are using diagnostic testing using gold nanoparticle as a biomarker. Nanoviricides, drug called nanoviricides designed to attack the only virus particles they're working on. Nanomedia on targeted drug delivery, nanoscience diagnostic testing system. There are so many, because if I will name, these are the some, because they are selective one, but this is the only data from the United States. I'm telling you, there are many companies throughout the world. They are working in the other Europe and Asia and other part of the world. So these are some selective companies. They are working, they're investing, they are developing their product, and some of the product are already, more than 100 products are already available in the market. So some of the commercially available nanomedicine for therapeutic purpose are like Doxil, that we know very well, that doxorubicin liposome is there, Donosome, Abraxane, Ambisome, Istrazob, Amant, Mesgec, Tricor, Triglide. These all are the nanomedicine-based products, which are FDA-approved products and are very much effective in the clinical management of the diseases. So, all right. So, how we develop the nanomedicine? What are the approaches? How we should go for it? So, there are three important aspects when we are going to develop a nanomedicine. Number one is material. What material you are choosing, and for what is the characteristics of that material, and what will be the process of manufacturing of nanoparticle. And after the development of nanoparticle, it will be characterized, and what are the regulatory aspect of these nanomedicines, whether it will be approved by the FDA or not, or other regulatory agency of the different part of the world. There are certain guidelines, they are being framed at the moment, so that it should be, because nanotechnology itself has certain disadvantages, like there may be nanotoxicology, because if the deposition in the wrong part of the body, it may lead to the too much problems for the patient. So safety is also important. So biodegradability of the nature. So when we talk about the material, we know that we can choose either polymer or lipids or metals or hybrid of these material type. So what are the factors that are important? Number one, ingredients that you have chosen for developing nanomedicine and their interaction with each other. Polymer to drug, lipid to drug to surfactant, and then there are some other. And what are the interaction with process component? Sometimes you are using the suppose you are preparing liposome, you are using hand shaking method, or you are using the ethanol injection method. There are n number of methods. So what are the process component that they are affecting? Then whatever the nanomedicine you develop, what is the performance of the desired characteristics of component in manufacturing conditions? If it is for parenteral route, whether it is sterilized or not, and how you will sterilize, and whether it will maintain its integrity. What are the acceptance of component at regulatory level, because you have used many solvent or other process, whether it is acceptable by regulatory bodies. What are the characteristics features that are retained in vivo? Because in vitro there are features, very good features, but after administration into the body, that nanomedicine will retain that or not, and whether it will perform the executed task or not. What is the in-process and storage stability of the nanomedicine? Because it may lose during this time, so stability is important. Reproducibility is important, because the one time you are preparing the nanoparticle, it is 100 nanometers. Second time you are preparing, it is 200 nanometers, and third time it is 300 nanometers. So there is no reproducibility of particle size, or similarly with the drug loading capacity and other features, in fact. So whether it is reproducible, or method process is reproducible or not, and what are the scale-up transfer feasibilities and capabilities of your system, whether it can be transferred to the industry from lab, that is very much important. So we have to understand all these at a time. So you can use n number of material, organic, inorganic, like metal, particle, polymer, and lipid. And there are several techniques for developing the nanomedicine, because each material type has their different characteristics, and according to that you have to choose the method. If you ask what is the universal method, I will say there is no universal method. Material is different, so process of developing nanomedicine is different. So there are different methods, different materials, and that's why there is a variation in the performance. So there is no universal method for developing a nanomedicine with help of polymer. If you say that there are nanoprecipitation, salting out, supercritical fluid technology, extrusion, microfluidizer, ionic gelation method, and each has their advantage and limitations. For example, nanoprecipitation develops the small particle size with low cost. But there are limitations like difficulties in containing and controlling the particle growth, applicability to lipophilic drug only. So each method has their advantage and their limitations. So you have to choose among the benefit and risk ratio. What is your intention and how you are going with that. So similarly, the lipid particles are also working, and that they are developing in the same direction. If you are high pressure homogenization, microemulsion, solvent emulsification, evaporation, membrane contact method, ultrasonication method. So every method has their advantage and limitations. You can choose accordingly. The question is, what are the challenges for the nanomedicine? Some of them I have already discussed, but if we talk about a proper mature nanomedicine, it requires the ability to build the structure and devices with atomic precision, because nanomedicine is very, very defined and very, very focused in this direction, and hence molecular nanotechnology and molecular manufacturing are the key enabling technology for this nanomedicine. So shape and size is important. Biocompatibility is important. What is the powering capacity, that is important. Whether it can communicate properly with the cells, that is our objective, and whether it can navigate it to the particular target site where the treatment is required. So all this is important in the one direction, and these are some of the challenge. One more important thing when we are developing, suppose nanomedicine, this is very practical aspect of the developing and challenge in the nanomedicine. When you are developing the same nanoparticle with the same drug, but the release profile is different, because it is very important to get a reproducible drug release profile from the nanoparticle every time. And it is required to establish the batch to batch uniformity in the scale-up process. Second is quality performance by in vitro and in vivo performance. So how you will manage if you are developing the generic nanomedicine or nanosimilars? So whether you can achieve the same quality performance by IBC, is it possible? Because this is a big challenge for FDA nowadays, because the patents are being expired for the existing nanomedicines, and the people are developing, the pharmaceutical companies are eager to develop the generics of the version of them same, but they are not capable of achieving that. So a lot of challenges are there, and after crossing all barriers, there is one product reaches into the market. So there are factors like therapeutic need is there or not. That is the first important. Market demand is there or not. Research and development, because it needs a lot of time and money and resources consumption. So a lot of investment is there. What are the production steps? Whether it is okay with these production steps in the large scale. Scale of feasibility is there, yes or no. Clinical trial successful, yes or no. And what are the regulatory issues, whether it is approved or not. So there are so many barriers. Afterwards, only one nanomedicine product reaches to the market. And there are these problems. So what are the problems with the scale of feasibilities? Usually we should select the material which is generally regarded as safe and having GRAS status. So it is more beneficial if we are trying to get the scale of feasibility easily. Also, we must understand the toxicological features associated with the size and shape of the nanoparticle. And in vivo biodegradability of nanocrystal is also important, because once they are administered in the body, they will retain for a long period of time. And whether they will be properly degraded by the body and safely they are metabolites are removed from the body, excreted from the body, that is very, very important. If it retain in the body, it will deposit in the brain or small capillaries, they may create a lot of issues, and there may be a problem. So we have to understand these points also, and then we have to make the balancing of multi-component system at large scale production, because when we are going to make a big batch of the same system, we have to understand, because there are involvement of multi-component in the production process. So every step should be developing the desired characteristics in the nanoparticle. So selection of the nanoparticle production method is very, very important, because it saves the time during pilot batch production from scale-up point of view. This is very important. Otherwise, you may miss that point. For example, if we understand a study in a comparative study of ibuprofen loaded nanoparticle, it was found that the nanoprecipitation method took less time, only 2 hours, whereas emulsion based method takes 3 hours of nanoparticle production. And this 1 hour of extra time that is in the method of production and without improving any other quality, it is the big loss for the manufacturing company. So we have to select the proper method so that we can save more time and do the rigorous optimization of nanomedicine with less cost, I will say. So this was all about the nanomedicine and its history and progress. Now there is one topic on which at the moment I am looking for, because we know that there is a huge market on clinical oncology in the nanomedicine. So this is on the treatment and therapeutic point of view, whether we can work on the prevention of these diseases with the help of the herbal products. So one concept is there that is called nano-chemo prevention. So we better to prevent the cancer, not to treat the cancer. So with the help of nanotechnology, with these nanomedicines and nanoparticles, whether we can deliver the plant products so that they can prevent the cancer and the progression of the cancer, and they can prevent. So this is the merger of your ancient knowledge with the cutting edge technology. So you know very well that these are the plants, they are being used by the many since ancient time for this kind of purpose. But if because we know that they have low bioavailability issues, so if we can deliver them with the help of nanoparticle, and so that they are more easily taken up, and precisely they are working as a nano-chemo prevention agent. So we know that plants of natural origins are there and they have applicability in carcinogenesis. But problem is they are very limited effective due to their physicochemical and biological barriers, like a flux bahut jaldi ho jata hai. So what may be the effective approach? We can use the nanoparticle for these delivery using different type of biomaterials like polymers. We know that PCLP, LGA, alginate, chitosan, PEG, ultrazit, lipids, gelatin, selenium, etc., etc. We can also make them very selective for targeting purpose using certain ligands on the nanoparticle, and we can utilize advantage of EPR effect, etc. If there is any chance of developing tumor or itself, they can fenestrate the junctions of capillaries easily. And when we are taking fruits, etc., yes, they are working. These materials are, these plant materials are reaching to our body, but their bioavailability is very less. So whatever we are eating, it is not 100% absorbed. So there is a no control and measure of intake, and depends upon the food habits. Usually, sometimes people take more fruit, sometimes they take no fruit. So there is no proper intake. Genetic variations are also there among the people and has poor viability. So with the help of these things, if we can conceptualize and we can develop the more effective herbal nanoparticles with the herbal drugs, they are more stable and have with high payload with controlled release properties, also capable of targeting and improved distribution in the body. And there are wide range of biomaterial options are available so that you can prepare and you can get the improved pharmacological activity overall. So this is objective of this technology, and this area is very important. And also the prevention, we are more focused on prevention than this cure. So this is a requirement of the future. So I have recently written a book in this area, as Dr. Vinod told, and we have compiled some of the information in this book on the what are the possible common molecules and what are the technology existing available, how we can go with that, and what are the regulatory guidelines for the herbal products, and how we can maintain what are the quality controls and other areas where we are working upon. Because I am working in a university which is more focused on the herbal estate and herbal area, so that's why we are working. Now there is one question from for the future point of view, that okay, this was all history, and okay, from 1900 from 1900 to 2000, we have made a journey so far. And 2000 onwards, what are the novel concept or the upcoming concepts on which the people are working upon in this area of nanobiotechnology and nanomedicine? So as I told you, number one, that people are working and the industries are working in developing personalized medicine, so that individual person, we can design and construct the monoclonal antibodies for one particular person, and we make the customized medicine for the same, so that there are no side effect or and better treatment. Second is nanotheragnostics. So nanotheragnostic, the people are working in both single nanoparticle having diagnosis as well as therapy. So the people are working in the direction of developing that same nanoparticle can reach to the site, and there we can visualize, okay, it reaches to the site, and then only we trigger the release of the drug, which is having the capacity of treatment. So simultaneously working in both the direction, diagnosis as well as therapy. So nanotheragnostics people are working nowadays, because we know there are so many requirement of regenerative medicine and tissue engineering. So nanotechnology is also being implicated for these areas where people are looking for the better treatment and more precise treatment in the developing tissue engineering, artificial tissue development, etc. 3D printed nanomedicine, that is the upcoming concept, because already 3D printed pharmaceuticals are being coming up, and with the help of this technology, more precise, more accurate nanoparticle or the delivery system can be prepared with the help of desired surface texture, and the characteristic size and everything is better controlled, because it is based on the artificial intelligence and the data provided by us. So selective polymers are being used to deposit a small nanoparticle or microparticles and advanced drug delivery system. So 3D printed technology is upcoming, and for developing proper characteristics and designed therapeutic parameters and physiological parameters, the optimization of nanomedicine development, not trial and error experimentation. The quality by design is being approached, and people are using the different optimization technology and factorial designs to get our desired features in the product, so that we can understand with the less number of experiment our desired quality characteristics, target product profile can be achieved in the nanoparticle and nanomedicines. Next area which is coming this era is nanosimilars, because as I told you that a patent are being expired for the nanopharmaceuticals, which was done long before, 20 years, more than 20 years before. So patents are expired or being expired, and the nanosimilars are being developed, means the generic version of nanopharmaceuticals are being developed. For example, Doxil patent expired, then Lipodox is being developed by Sun Pharma, and similarly, Abraxane and all other Myoset, and these are being expired soon. So the already nanosimilar, the industries are working in developing generic nanopharmaceuticals. Then people are also looking for nano devices for gene therapy, more accurate gene therapy, and those nano devices which can be scale up and which can be transferred from the lab to the market. So that kind of nano devices are being used with the help of pharmacogenomics or the proteomics and genomics knowledge. Nano robots, they are the small tiny particles. As I told you, that ideal concept was proposed. Still, nano devices are being developed, which have all kind of, I must say nano bioelectronics, having the sensor, having the capacity to release the drug, and or the making a microsurgery or nanosurgery at one particular small platform, like if there is an blockage in the cardiac artery, and small nano robots can go and remove the clot, and it can clear the obstruction for the blood flow, and the patient can feel better. So that kind of nano robots are being designed, which are controlled by the computer, and for the micro and nano surgery at very small level, even for brain surgery, etc. also. So these were the references that I have used in this presentation, and I hope I have tried my level best to little bit explain what is the nanotechnology and nanomedicine and nanobiotechnology, and what is the future of it. And this is my university, so I personally invite all of you to come and visit the beauty of the location, this Amarkantak region. And thank you so much. Here I wind up my slide. If there is any query, you can ask.
Thank you, sir. Thank you. It was very wonderful talks.
Thank you.
So, participant, if you have any questions, you can ask directly, or you can put your question on the chat box.
Hello, sir. In fact, you are here, and the cases of the corona are on rise again. So I would like you to throw some light on that, how these nanomedicines, how could you see these in prevention, diagnosis, treatment of the corona?
Hello.
Hello.
Can you repeat the question, please?
What Dr. Shah was asking that how do you see nanomedicine in prevention, diagnosis, and treatment of corona?
Yes. That is very true. Because we know that all there are several areas where the nano vaccines, nanoparticle based vaccines, particularly. For that reason, I must say 2 points are there. Number one is therapeutics. So we are like remdesivir injection is already available. So if we can more precisely design that remdesivir loaded nanoparticles, and we can understand the spike proteins which are available on the corona surface, and we can utilize in smart way so that wherever the virus particle is there, we can attack on it. Secondary, because we know the deposition of these this coronavirus is more on respiratory tract, so we can also utilize in future. I must say, because it takes time, but through the nose to the respiratory system delivery, we can also utilize this nanotechnology. And third, we know that this boosting of immune response with the help of nanoparticle, just like we have done previously with the influenza vaccine. So we can, this is a more like the same virus of the same size, and we can also boost the immunity. And in this direction, the work is going on. Nano emulsion are being utilized in one of the clinical trials and the vaccine. So nanotechnology itself is having certain advantage in this direction, and it's working.
Thank you.
Thank you.
Anyone else? Participant? Anyone else?
Hello, sir.
Yes.
Hi, sir. This is Dr. Binda Sharan, sir.
Yeah.
I'm having a question, sir. If you are having a drug which is having a based on the lipophilicity property of the drug, which method preparation will be useful to focus on the nanorobotic application?
Your voice is having some echo. I'm not getting your question clearly.
Is it okay now, sir?
There is so much echo actually. Sir, if
Is
you
it
heard,
okay now, sir?
yeah. If you can repeat, sir, Vinod, sir.
Hello, sir. Is it okay now? Still echo, sir. Sir, this
Hello.
is
There is
based
echo.
on the lipophilicity property of the drug, which
Networks पर टाइप कर सकते हैं.
हाँ.
सर प्रेजेंटेशन को बंद कर दीजिए।
All right.
So participant, you can type your question on chat box also.
Actually, nano robotic applications are different. Actually, what I am telling is nano robotic applications are for surgery purpose. Actually, if you are preparing nanoparticle of some lipophilic drug, uh, obviously our intention is to upload the maximum possible drug into the nanoparticle system. So if it is more lipophilic and there is no constant, then I will suggest go with the lipid nanoparticle, because the lipophilic drug has very much affinity for the lipid itself. So better to use that lipid nanoparticles, maybe solid lipid, maybe lipidic carriers, or maybe your liposomes. So they have better bioavailability. And since liposome already have the commercial aspect, so I will suggest if there is no problem, you can go with the liposome, because scale of feasibility is settled and there are FDA guidelines also. So if you are working in that direction, it will be easy for you to commercialize your product. But nano robotic means it is for surgery purpose, not for the drug delivery purpose. So that is what I am. Thank you.
Yes, Bhavisa madam.
Yes, good morning, sir. Good morning, Rishi, sir. Sir, I am Bhavisha Patel, working as an associate professor in a school of pharmacy itself. Sir, one question is there. Can we convert the herbal nanomaterial into coating material if it is having an antiviral activity? So
Yes, we can. Because we know that in tribal people, like the local people from ancient time, they were knowing that okay, these drugs are used for this kind of treatment. Though they were not familiar with virus and fungus and bacteria, but they know that if this kind of problem is there, we use these kind of plant-based product. So those plant-based product can be converted into the nanomedicine product. That is what I have mentioned in this nanopharmaceuticals or herbal nanomedicine, if I say. So there are plenty of opportunities. You can convert them. Either you can encapsulate, or if you have isolated compound, then you can directly upload. If you have plant extract, then you can also encapsulate into the liposome and other vesicular type of system. So there are possibility. Yes, we can do it, and also people are working in the direction. Like I can tell you, this encapsulation of the essential oils and aromatherapy, that is also being implicated with the help of this nanotechnology, nanoparticles.
But I want to convert it into the coating material, is like in a mask and everything.
Yeah, that can be done. Nano particle that you coated on you can use any metallic surface, because metals like copper and others are also active against the antivirus, this coronavirus, if I say. I'm right, because you are asking like that. And you can also coat these herbal extract or herbal compound if you know their antiviral properties. So altogether, this spray will be very much useful in the mask for prevention. Yes, we can do it.
Okay. Thank you very much, sir.
Thank you.
Okay. Any more? Anyone else? Okay. Thank you, sir. So it was a very wonderful talk, and our participants definitely get benefited with this talk. So we would like to express our gratitude on the form of a e-certificate, sir. Please accept it.
Thank you so much. Thank you so much, Dr. Binod. It was nice talking to all of you. It was good day, good interaction.
Please accept it.
Thank you so much. Thank you. Thank you. Thank you. I hope next time we will meet physically and it will be
Definitely, sir. Definitely.
one by one. Because of the time limit and all other areas, so I was trying to compare with the introduction part only, not the application part. So thank you.
Lata ma'am, would you like to add something? Ma'am, your voice is not coming.
Hello, Rishi sir. It was really very nice and wonderful session.
Thank you so much, ma'am.
Very
Thank
lot.
you.
You have touched all the aspects of nanotechnology and nanobiotechnology. The place. Thank you. Thank you for.
Thank you.
Yes, sir.
Thank you so much, ma'am. Thank you. We will meet you. Thank you.
Okay. Thank you, sir. So with this, we end the webinar.
Right.
And I will share a Google form link for the e-certificate. So please fill this form to get e-certificate. Thank you, sir.
Okay. Thank you so much, Dr. Vinod. May I leave now? Thank you.
Yes, sir. Yes, sir.
Thank you. Thank you so much. Thank you. Bye. Bye.